It's a
It's a
pleasure for
pleasure
me
for me
to
to
introduce
introduce
today's
today's
special
special
lecture
lecture
uh,
uh,
speaker,
speaker,
Dr.
Dr.
Ritesh
Ritesh
Kumar.
Kumar.
I have a
I have
very
a
long
very long
association with
association with
Ritesh.
Ritesh.
I
I
think
think
approximately two
approximately two
decades of
decades of
years.
years.
Dr.
Dr.
Ritesh Kumar
Ritesh
has
Kumar
done
has
his
done his
PhD
PhD
from
from
Korea,
Korea,
and then
and
he
then he
moved to
moved to
postdoc
postdoc
in
in
USA,
USA,
and he
and
has
he
done
has
two
done two
postdocs,
postdocs.
one at
One at
University of
University of
Missouri
Missouri
and
and
another
another
at
at
University of
University of
Minnesota.
Minnesota.
And in
And
both
in
the
both the
postdocs, he
postdocs,
has
he has
worked
worked
upon,
upon
improving the
improving the
nutritional
nutritional
quality of
quality of
soybean.
soybean.
And
And
recently, he
recently,
has
he has
joined a
joined a
company,
company,
Ball
Ball
Phenotypes of
Phenotypes of
Horticultural
Horticultural
Crops.
Crops.
So, with
So
this,
with
I
this, I
thank
thank
Dr.
Dr.
Ritesh
Ritesh
and
and
invite him
invite
for
him for
delivering his
delivering his
lecture.
lecture.
Thanks, Dr. Neil. It's a great pleasure to be part of this workshop, and thanks for the invitation. So, good evening, everyone. As you all have the all brainstorming session so far in this workshop, and I would like to present my previous postdoc work, basically upon soybean seed composition and how we modulate them and what are the challenges during that modulation. So, wait a minute, I will highlight my marker. Okay, doesn't matter. So, first, let's have a small introduction. As a plant scientist, we have multiple future challenges, and we will go one by one for each of them. The first is, let's say, population. As you may know, like by 2050, we need, and India will be the major contribution in that population. Well, so we need a high yielding crops which can feed our future and present generations. Second is nutrition and taste. So we need balanced amino acids, vitamins, and allergen-free crops. So as you see, the climate is changing drastically. You can have heavy rains, droughts, and multiple freezing conditions. So you need a crops which can combat these climatic variations. At the same time, as human activity is expanding, plant diversity is limited. So we need to conserve our plant diversity for our future generations. We are coming up with multiple chemicals or herbicides or pesticides increasing plant seed pests, insects or diseases. We need the crops which can stand by or even counter these modifications by these smart insects or pests. So we thank solution which with which we can feed our next generations. So today's talk I will focus upon the challenges about nutrition and taste, and for that I will talk about soybeans. So as you know, the genetical name glycine max here. So why soybean? As soybean is the world's most important seed legume. Why I'm telling that? Because in so here if you see its production, so Brazil is in the top, followed by US. India is in the third or fifth position total world, but still it's a significant number if compared to the other world countries. So in case of soybean growth in US, it is the second largest. Corn sometimes leads, so it's fluctuate. Sometimes soybean is on the top, sometimes corn is on the top. So they are first and second most largest crops grown in US. So if we talk about different states in US, so I work at University of Minnesota, where around 7 million hectares annually were grown. Second state where I work is Missouri, that is also among top 10. And the state where I'm currently living, but I'm not working with soybean, is most highly productive in soybean. If you talk about India, you see only two states, Madhya Pradesh and Maharashtra. Most is the Madhya Pradesh are the two major contributor. You can see some faint green patches all around India, but still the production is very low. So major contribution are just two states. So and in India, the soybean season start early June and that's the harvesting in October. And here is the soybean yield in India, so which you can see it's over the 10 year. It's an increasing trend, so that's a good indication here. So if we see about soybean uses, we know about food products like tofu, soy meat, sprouts, soy sauce, or yogurt, soy milk, and mostly the soybean oil. But main consumption of soybean is for the animal feed and multiple industrial uses ranging from tires, lubricants, biodiesel is a peak now, paints, plastics, and good quality adhesives. So if we talk average seed composition of soybean, it's divided into two parts. First is oil, and main is the meal. So 80% is meal, 20% is oil. If we further go down, so meal is composed of 40% protein, 18% insoluble sugar, 8% soluble sugars, 4% minerals, and 10% moisture. If we talk about uses, 97% of animal feed is used for 97%, and 3% is just for food products by human consumption. If we further characterize oil, it constitutes of 5 major fatty acids, namely linoleic acid, oleic acid, linolenic acid, stearic acid, and palmitic acid. And if we talk about uses, 68% used for food consumption, 25% for biofuel, and 7% for the industrial uses. So as you may know, that soybeans are among the big 9 food allergens. So what does this mean? So, and why should we worry about food allergy? So I'm talking about US. There are like 15 million people who have a food allergy and have a serious medical conditions. And every 3 minutes, you can see a person going to emergency due to the food allergy. And this number is increasing. Now we have 50% increase in the food allergy cases among children. And you can find one children is affected among 13 in the schools having food allergy. Although treatments are being developed, there are no cure for the food allergies. But that doesn't mean you should give up eating soybeans. Because why I'm telling that, because just 1 to 5% per 1,000% is the effective or the allergy rate for a soybean. So soybean is highly safe to eat. So if we talk about sorry, something goes wrong. Okay. So if we talk about plant proper seed germination and the development of young seedlings, you can see they need a source of nitrogen, carbon, and sulfur, and which comes from seed storage proteins. And if you sediment this total protein of soybean, they are characterized into different fractions, starting from 2s, 7s, 11s, and 15s. And major fraction is composed by glycinins, that is 85%. In case of 11s, then 7s, that is 35%, that is beta-conglycinins, and mostly these are the dominant one in this fraction. 2s is composed of Bowman brick inhibitors and kinase trypsin inhibitors. And 5% is the polymer of different soy proteins. So these two are the major dominant storage protein, beta-conglycinin. I may call them 7s globulin in my further slides, and glycinins, which are also known as 11s globulins. So these are the different soybean allergen, and these are different properties. Major dominant storage protein, each of them, glycinin. What I may call them 7s globulin in my further slides, and glycinins, which are also known as 11s globulins. I have some restrictions from my previous labs, soybean allergen, and I'm walking through the properties, the names and everything, each of them, and what I may call them. So I have to find go for further allergen families. So I have glycinins, which are family A, 11, C, and D. So these are confidential. I have some restrictions from my previous labs, soybean allergen, and I'm walking through the properties, the names and everything, each of them, and what I may call them. So I have to find go for further allergen families. So I have glycinins, which are family A, 11, C, and D. Let's talk about irritating tools to make. I have some restrictions from my previous labs, soybean allergen, and I'm walking through the properties, the names and everything, each of them, and what I may call them. So I have to find go for targeted 5 different families of soybean allergen or storage protein family. And we are using multiplex card RNA. I will not go in any detail for Cas9 because from last few days you guys are hearing about all these things. So this is a normal agrobacterium-mediated whole plant transmission for soybean, starting from a cotyledon, seed cotyledon, and resulting into callus and different fruiting and shooting stages. And finally, we get a plant, and we screen the T0 generations with the single sequencing and now also we are using Illumina sequencing. And finally, we get a T0 sequence and we sort out which plants have mutations and they are transgenic. We move them to the next generations and so on. We have to get the transgene-free plants in the next generation, so we segregate out those populations, and finally we go with the plants which have just mutation and no transgene intact in it. The first family which I'm talking about is a glycinin. It's also called 11S. It has 5 major members ranging from glycinin 1 to glycinin 5. You can say GAI1 to GAI5. So once they make a protein, they have acidic and basic chains. You can see A10 or AB. So this is acidic and B stand for basic chain. So what these proteins goes under post-translational modification, and they make a hexamer, as you can see here. They're making a very nice different combinations. And this is one of the structure for A3B4, this 3D structure. So these glycinin are severe soybean allergens. So they are so dominant, they are used as a marker for soy detection in food or blood. So these are different amino strips. And glycine consumption in animals can lead to intestinal damage, oxidative stress, growth retardation, dyspepsia, allergic diarrhea, and also some IgE-mediated responses, immune responses. So what we did, we made a sequential glycinin mutants using CRISPR-Cas9. So first we made a single knockout, double knockout, and triple knockout. Triple mutant. I'm calling it a triple mutant because third gene which we tried to knock out was not knocked out, but it's knocked out. So it's a triple mutant. It was in-frame mutant. And we confirmed the mutations by Sanger sequencing. So as you can see, the total glycine content was reduced in a serial manner or as dose-dependent manner with the mutations increasing. So then we did a total seed protein mix, the whole seed protein mix for these mutants and a control. That is Williams 82 here. So gray bars are the Williams 82, that is the control, and green is the single knockout. So as you can see among these 5 genes, this guy 5 is significantly knocked out in case of the single knockout. If we talk about double knockout, again, the gray is the Williams 82 control, and the yellow is the mutant. So 2 genes, 2 proteins which are affected are the GAI3 and the GAI5. They are significantly knocked out. So another mutant that is I call triple mutant. So as you can see, among GAI3 and GAI5, GAI4 was the target, which was significantly reduced. But along with this, other 2 proteins, GAI1 and GAI2, were also reduced. We don't know exactly what's the mechanism going on. We didn't see any mutation in these proteins, but there may be something post-translational modifications or some upstream things going on. So that need to be taken care of. The next family which we targeted is beta-conglycin and 7S. So it has 3 subunits, alpha, alpha prime, and a beta subunit. And they make a trimer. So you can get 8 different type of primers with the combinations with these different subunits. This is one of the example for homo trimer of beta subunit for beta-conglycin. And the same like glycine, they are also dominant soil allergen used as a soybean marker for detecting soil in foods or bloods. And they can also cause intestinal damage, growth retardation, increased histamine and Ig levels. So mostly these are different immune responses which are resulted with the consumption of this protein. So we target all these three subunits in this mutant using CRISPR-Cas9 to confirm the mutations by single sequencing. So these proteins are so dominant. So if you run a protein gel, you can see very clearly on the STS-based gel. And this is the bird, is a wild type. This is a different genotype. Earlier we talked about Williams 82. So this is alpha. This is alpha prime subunit. This is a single sugar binding protein, and this is the beta subunits. And these are the conglycine knockouts. So you can find these units are simply missing in these mutants. That means they are completely knocked out. And further, we quantified these with the proteomic study. So these are the peptide counts. Green bar is the control, and yellow is the conglycine knockout. So as you can see, these all 6 proteins are significantly knocked out, or you cannot see in the these graphs. So that means our CRISPR-Cas9 is efficiently working, and we were able to knock out 6 different proteins at one time. That's an efficient gene multiplex gene editing for soybean. Another family is allergen A, and it has also come up with 3 different set of mutants. So it's not like we target in such a way, but it's a random match. During segregation, we found just 1 gene knockout, 2 genes knockout, and then 3 genes knockout. So it's a segregation population of a single construct. So first, we confirm it with a Western blot using as an A antibody. So as you can see, its traction is showing. It's reduced significantly in case of the triple knockout compared to the single knockout. And we confirm these results with the whole seed proteomics with the DFSF method. And this is the control. Allergen A single knockout is significantly reduced. Double knockout is A1 and A3 are knocked out. And in case of triple knockout, all 3 versions of allergen A are knocked out. And if we talk about percentage, in control is 6.12%, and that is finally reduced to 0.29%. That's just some background. So this is the percentage which has finally got after CRISPR-Cas9 Act. So another mutant which we made is a triple null mutant. So why is it triple null? Because we use CRISPR-Cas9 acting upon 3 different families of the allergens. That is allergen A1, allergen B, and allergen C. So similarly, we confirm allergen A1 with allergen A antibody. So I will tell you, so before we going to this biochemical assays, we confirm the mutations with the Sanger sequencing and only took the homozygous lines for these assays. So these are 2 different CRISPR-Cas9 lines which we shown here. The allergen A is barely detectable in case of the CRISPR mutants. And also we confirm those with the proteomics results, and you can see it's significantly reduced here. Second is allergen B. So that's the dominant protein. You can see that on the gel here. And this is the wild type. You can see that band very clearly, but that band is missing in case of the mutants. And further, we confirm that with the proteomic study. You can see that it completely reduced. Another part is allergen C. This is just 2D gel. You can see this is allergen C. And we know what the bands and dominancy, and that is missing in case of the mutant lines. So as you can see, the green is represented of this CRISPR-Cas9. So that is missing here. So, and we also verified those results with the proteomic study, and this is highly reduced as comparison to the wild type in the yellow clay. So mostly these all mutants which we produce are due to the frame shift mutation, early stop codon, or totally different proteins. So that protein is no functional anymore. So take home message from here is we were able to knock out several allergens or anti-nutritional proteins using CRISPR-Cas9. So first is glycine family knockouts.
conglycinin family knockouts, then we made allergen A family knockouts, and triple A knockout. So you might be thinking problem is solved, but not. That is the real struggle. So why I'm calling that? Because the protein rebalancing in soybean. And I'll tell you how it hindered this process of making allergen-free soybeans. So for that, we should know what is protein rebalancing first. So let's see, this is an instance where these are the two dominant proteins, 7S globulins and 11S. So mostly 11 is in a more a higher percentage as comparison to the 7S, but this percentage may vary due to the environmental factors or genotype to genotype. Let's instance, if you reduce the 7S globulins, this loss is compensated by the 11S globulins. So 11S globulins go up or other storage proteins goes up to compensate this loss. Similarly, if you reduce 11S globulins, this loss is compensated by increase in the 7S globulins or other storage proteins. So this compensation of the loss of one protein by another protein is known as protein rebalancing. So why understanding protein rebalancing in soybean is important? Because soybean seed protein has two output rate. Let's see, that's total protein, that is the quantity of the protein measured by total nitrogen. And the seed proteome, that's the quality of protein depending upon the amino acids concentrations. So if we tend to increase either of them, either the total protein or quality of the protein, that's specific amino acids, the major hindrance here is protein rebalancing. So for instance, poultry need a methionine-rich diet. And in case of soybean, although it is highly protein rich, the methionine content is very low. So what people are doing is using less expensive alternates, that's artificial methionine, which once upon a time was banned by USDA for human consumption, but now it's again in the market with some limited doses. So we need a methionine-rich soybean, and people are working upon it from decades. So this using a transgenic approaches where we can induct the foreign proteins with high methionine codons. What we expected is the expected outcome of multiple foreign bodies which have high methionine codons. What real scenario is this? Just as few attempts which are like barely successful. So here, Valen is protein rebalancing. And we want to make this protein rebalancing as a hero, and how we can make that by understanding the mechanism. What are the key regulators which controls the protein rebalancing, and how we can utilize those in the future. So once we have a control over protein rebalancing, we can optimize protein composition. We can have a high protein meal. We can have a desired amino acid composition. Let's say high methionine for poultry or swine feed. At the same time, we can also have a high quality protein, plant protein for human consumption with all essential amino acids, or we can mainly work upon allergen-free soybeans. So we will have the quality food, we will have the desired meal, and multiple industrial uses once we control the protein rebalancing. So this is a part of my first postdoc here, where we found that this protein rebalancing is a dose-dependent, works in a dose-dependent manner. So how it happens? So say we targeted three different members of 7S globulin family, alpha, alpha prime, and beta. And red and blue are two different guide RNAs which acted upon different positions in these all 5 genes. Although alpha subunit has 2 base pair difference between the guide RNA, but it still works. So that is a simple Cas9 construct which we used for this mutants. And this guy here, it worked very efficiently. So what we found during segregation, different 3 combinations of mutant. One is a single knockout. This is the wild type here. You can see these are all units, alpha, alpha prime, alpha, and a beta subunit. In case of single knockout, this beta subunit is totally missing. And we confirmed it at the mRNA level by qRT-PCR and also at the protein level by whole seed protein mix. So these were significantly reduced as comparison to the control. Second is a double knockout. You can see this alpha prime is missing in all the mutants, and beta subunit is also missing in these mutants. So this is alpha prime and beta subunit double knockout. We confirmed this at the mRNA level by qRT-PCR here first, and then with the double knockout at the protein level, you can see alpha prime and beta were significantly knocked down. The triple knockout, you can see alpha prime, alpha, and beta subunit. All three subunits are missing in case of the mutants. And we confirm similarly at the mRNA level, all three are gone, and also at the protein level, all three are significantly reduced. We were able to make these three different versions of mutant in the same family to have more in-depth study for the protein rebalancing. And they were reduced in a serial manner. That is also good for us to consider multiple factors in the future. So when we check the crude oil and the total protein, they are same, they remain unchanged. That means even if you are taking out the big chunk of proteins out of the proteome, but total protein is same. So that means there is happening protein rebalance. So when we did a proteomic study in this mutants along with the Magellan, that's a wild type, another genotype, we found the targets which we use is conglycine is 14.47% in wild type. That was reduced to 2.4% in case of the triple mutant in a serial manner. And the protein which rebalanced is mainly is a glycinate. So which was 20% in the wild type was around 28% in the triple knockout, and that also increased in a dose-dependent manner. So more is the conglycine 7S goes down, more of the glycine goes up. Then we want to see whether this is happening at any transcript level or not. So people have reported previously like this is a post-transcript modification, but we still want to confirm that. But to our surprise, we see some significant differential expression of these genes at different developmental stages, and that's also at very specific developmental stages. So we use all this R5. I'll show you the diagram in this. So we can see this mRNA expression is increasing in a serial fashion. It's a dose-dependent manner increase in the gene expression, and which is similarly found in other genes too for these two stages. So what are these stages? So this is R5.1. So this is a fresh weight, 25 to 50 mg. 75 to 100 mg is R5.2, and so on. So R6 is the final stage to 300 to 400 mg for the fresh weight. So our froze in the field, and then to back to the laboratory, RNA was taken out, and from that we made a cDNA and confirmed these experiments. So here we found as multiplexed involved in protein rebalancing in a dose-dependent manner. That is very important finding here. So with the increase in the CRISPR-Cas9 induced mutations, we found conglycine mRNA is going down. White, blue is the high and white is low here. So similarly, conglycine proteins were reduced. On the same time, 11S and other storage protein gene expression was increased, and the storage proteins were increased. Not only 11S other storage proteins, we also find some stress responsive proteins or proteins related to translation machinery and translocation were also increased in this study in a dose-dependent manner. Similarly, we go in more deep, study the protein rebalancing at University of Minnesota, where we use a multiomics approach to solve the mystery of protein rebalancing in soybean. And we have three different set of mutants to study that. Set 1 is conglycine 7S, set 2 is Largene family, and set 3, where we targeted three different families of the soybean allergens. So, and we did an RNA-seq and a whole seed proteomics to get into more details about this protein rebalancing function. So here is the Largen A, where you can see we have a single, double, and triple knockout. The total protein remained the same. This is the Largen A, which was targeted as comparison to the K control. These were significantly reduced in a serial manner. At the same time, you can see increase in the 11S and 7S globulins, and also Largen B and Largen D. So different allergens are increasing when you're reducing the one allergen. And also, to our interest, when we, you see it's a single knockout, it's a double knockout, and it's triple knockout. So when we check the protease inhibition activity of these mutants, we we expected the triple knockout should have the least protease inhibition, but it was not the case. Only the single knockout was the least, and then it started increasing with the mutation. Why? Because that loss was compensated by another protease inhibitor, that is allergen D1. You can see D2 and D3. So these are increased in the double and the triple mutants in a serial fashion. Another mutant where we targeted three different families, triple null. You can see here, this is the wild type, this is a triple null CRISPR mutant, no change in the protein level. So that's when protein rebalancing is happening. We targeted the red ones, allergen A, significantly reduced, allergen B totally missing, allergen C is also missing in case of the mutants. And what compensated that loss is conglycinin and glycinins. So in case of beta-conglycinin, that's another family which we target at University of Minnesota. You can see similar to the University of Missouri results. This was also reduced. And we found an increase in the glycinins, allergen A, and some 7S globulins. So these were the results which we found at the protein level. We see the clear doses compensation by other loss of one protein by another protein. So we tried to get the allergen-free proteins, but it's not a case. We were increasing another allergen at the same time. And also, we found that amino acid profile for these mutants is remain unchanged. That's meant that amino acid bulk is also rebalanced in case of mutants as compared to the wild type. So the first is the what, that's the control. Almost similar range, as compared to the wild type. So here, in case of conglycin knockout, this are rebalanced by glycinin and allergen A. Similarly, allergen A knockout, they were compensated by glycinin, allergen D, and allergen B. So that is a rebalancing at the protein level. Now, we want to see a more in-depth whole like transcriptome level, what's going on, what genes are controlling this phenomena. So we did a mRNA expression examination at different developmental stages, started with R5.1 to R6.2. And you don't need to focus upon, I will come to the simple slide here. So these are the different stages. We use the RNA sequencing, such an next generation sequencing, and we've targeted the beta-conglycin knockout, where the beta-conglycin were knocked out and they were compensated by glycinin. So first we see these are the genes which we targeted. So black bar is your control and the knockouts are in the orange. So you can see the blue shows the minimal expression in both stages, R6.1 and R6.2. That's completely knocked out here. But at the same time, this is the control for the glycinins, these proteins which compensated this loss. They're highly expressed. So that means this loss or this compensation is happening, started very early at the transcript level. That was previously it was a mystery. Similarly, it is for allergen A, triple knockout. These were the genes which we targeted, and you can see these are cleanly missing in case of the allergen A triple knockout. These three were targeted, and other discompensated by another protein like glycinins or some parts, even the family of the same member. So I will conclude that in next slide. So these are the three which we targeted. So now what we are doing, we are making a co-expression based network modules for the differentially expressed genes among different seed developmental stages, and we are looking like what's common going on in these different set of mutant during the development and during protein rebalancing. And we have enriched some specific genes. Let's say this meiotic sister chromatid. This is related to something epigenetical modifications, and some different sort of combinations of different genes which are working together to getting this happen. For instance, you can see this co-expression based module 1. specifically this stage R6.1, these genes are highly upregulated. So they are working together in a one fashion in all the mutants. So that is very important. We are finding some very important proteins here, which are responsible for the protein rebalancing, but we need further testing to really establish their function in this phenomena. So basically, we are looking something which is common among these two mutants. What are the common regulators or transcription factors or epigenetic player? And also at the same time, we are tracing epigenetical changes by nanopore, like what changes which we can see in these mutant in process of the protein rebalancing. An important aspect of this study, this started last year when I was in University of Minnesota. So we have now different sets of mutants, different families, different genetical background, somewhere it's the same genes, but different storage proteins. So as you can see, we have control and mutants for each of them. So Professor Thomas Clemente from University of Nebraska pitched the idea. So let's study the genotype versus environment effect on protein rebalancing. So we have a beautiful set of mutants from Professor Bob Stoker from University of Minnesota, and Bing Stacy from University of Missouri, along with Professor Ben Parrott from University of Georgia and Professor Nathan from South Carolina. So which agreed to spread these mutants in all 5 different locations across US. So last year we have good planting season and harvest. This year they are growing in the field, and we are expecting a quite significant amount of data from all these mutants. So take-home messages from this talk: gene editing tools are efficiently working in soybean to knock out multiple genes at a time. This is just an example. You can use these complex systems for any genes or multiple genes in soybean very efficiently. Protein rebalancing is a dose-dependent phenomenon that we came to know. Protein rebalancing starts at transcription level, which was a mystery earlier and also is a debatable question. So, and also we are considering a role of epigenetic players in protein rebalancing because our proteomics and transcriptome data show some elements which are responsible for these changes. And during these studies, we found that protein rebalancing and storage protein accumulation is affected by environmental conditions. So if you are growing one mutant in a northern zone and one in a southern zone, you can see the changes in the protein rebalancing patterns. So this is the normal season at a super lab in University of Minnesota. We start the planting in early May. You can see it started the germination. June, you can see some cotyledon coming out. So soybean go very fast. Like once you grow, you're done. In June, you see some tiny, but by July, they are mature plants and started flowering in the next few days. And you have good friends to chat in the field here. And by October, we get harvested. By December to April, it's all covered with snow. So that's a normal flow chart for the stopper lab. So after working 5.5 years with the soybeans, last year I moved to wall horticulture. That's my first job as a bioengineer. Here, if I talk about wall horticulture, it's a family-derived company. It's a 4th-generation family company founded in 1905 by George Wall. So he's a founder. And now we have Anna Wall is a 3rd generation, and Susan Wall is the 4th-generation leader of this company. And our motto, as you can see, is all the best and nothing else. So we are spread around 6 continents around the globe, and you can see we have different functional units, breeder and producers, central research and development, that is wall helix, where presently I'm sitting and working, and breeding locations, production, that is our farm and fresh seedlings. And then we have a distributor, young plant producers, and some other parts that's related with sales. So we also sell for our competitors. That is a unique function of our company. This is one of the beautiful garden just behind me. It's on a view for our customers in the coming few days. So how the wall company works, we have R&D for breeding and for bioengineering here, cell biology, we go to the seed or cutting production. Then we have distribution that resulted in young plant production, distribution again, and result in finished plant production, retailers, and then go to the consumer like us, the gardener. So WAL is an harmonized, you can say it's an umbrella of different sub-sister companies like Pan American Seeds, WAL Flower Plant, Darwin Perennials, WAL Seed, and WAL Helix. So these are multiple companies working together under one umbrella. So it's not a single company here. So I will share some of the things initially, like our motto is to color the world. So WAL is a global leader in horticulture, and we develop, produce, and sell plants around the world, including India. We have branches in Pune, and I met a person recently visited our in West Chicago here, and we deliver all the best and nothing else. That is a very supportive motto of the company. So these are the seeds of success. It's not for our company only, but even for the young professionals like our PhD students here, or even for your professional and also for your personal life. So never sacrifice the long term for the short term. Innovate in everything you do, and run to the hard stuff. Don't go for the easy stuff first. Okay, so run to the hard stuff. And mainly communicate. So communication is very important in all aspects of your life, for your professional or your personal life. Communicate. Whatever the situation, communicate. And always do the right thing. So presently I'm working at a WAL Helix. It has 5 different locations around the world, 3 in US, 1 in Netherlands, and 1 in the Thailand here. So WAL Helix is constituted of 4 different parts: tissue, culture, and cell biology, plant pathology, molecular marker, developmental development, and bioengineering. That is the team which I am part of. And then we have research operations. So if you go one by one, this is the building where I'm sitting, and it has a beautiful garden outside, you can see. And people can see us, and we can see the people from inside the classes all the time. It's all light open. So we have first tissue culture and cell biology lab, where we do the tissue culture production services. We conserve multiple plants, and also like the cell and reproductive biology happen. And at the same time, for the bioengineering process, we need the plant transformation. They help us in the plant transformation of multiple species which we are targeting. Very important part of our lab is plant pathology and diagnostics, which do the pathology diagnostic services for not only for our company, but other companies too. Breeder support and production research, regulatory pathology. So if you have to bring any species from outside US, or you are sending from US to other, you have to test them first before they go to the market or to our distributor. Proactive global pathology network, and these pathologists are really associated all around the world. All are different locations, they work together. Molecular marker development and bioengineering. Now we have the ability, and we have sequenced multiple species at our own house and assemble them, and that there are really important genetic resources for our research. So we have a genetic research and breeder support, bioinformatics and data sciences, and then bioengineering, which I am the part of. Research operations, so that is portfolio management, research infrastructure, safety and regulatory compliance, data management. So that is very important. If you have to launch any future products, you have to we need a research operation which for us to get them deregulated or established in the market. So as a bioengineer scientist here, we are creating excitement in the world of flowers. We are looking for extending shelf and base life, speed breeding, genomics, and data analytics. That is, we are doing assembly of different genotypes, different species, or different flower varieties here. We are also working upon new exciting flower color, shapes, and fragrances. Better plant habit, that's maybe branching or compactness, more efficient nutrient and water use, maybe that you can say like aerotic stress tolerant or biotic stress tolerant plants, high resistance key plant pathogen, that's a biotic stress tolerant. And as you may see, heard about the night blooming petunias. So that may be the one of the future products. No, that's from another company, but we are also thinking of working upon it. So you may heard about night blooming plants in future from our company. So these are some examples of the products which came from wall helix. None of them is bioengineered, but they are totally cell biology, or you can say like male sterile, or they fuse different combinations. So these are breeding based products, but they come with the help from the wall helix. So as you can see, trait improvement. So if we started from crossing and breeding, it takes more time, right? Specificity is low. But when we come to the mutation breeding, it has time is low, but still the specificity is high. But with the new technologies, with genome editing or transgenesis GM approach, we are very specific. So specificity is very high, and at the same time, time is low. Only time we take here is for deregulation of the crops. So once it's deregulated, it's open to the market. So as you know, these are some examples which recently people done with the ornamental plants like enhanced rose scent or dwarf bananas, or you can see change in the leaf pigments, red or glowing in the dark, purple color, or pathogen resistance, or flower longevity. These are things people are doing, and we are also following the similar pathways or different gene regulations which we can work with. Working with academia and industry, I came to know is a little bit different. We have to pick the genes very carefully so that no one can work, or like no one have work on it, or no one can take responsibility of it in the future. So those are the difference which I feel here. So we're working with the gene editing, gene modification, and induced mutation presently to make the colorful word. So our motto is color the word. So we are making the word more colorful with these new techniques. With that, first I would like to thank the Superlab, mainly Professor Bob Stuper here, and our collaborator. The quantitative part is done with Dr. Vishnu Ramaswamy, and also the Stacy Lab, especially Professor Bing Stacy and Gary Stacy, for this opportunity to work on such interesting projects. And finally, what is common in these two labs is soybean. And with that, thanks, and I would like to happy to have some questions. Thank you.
Anybody has any question? No. If any of the online participants has any question, please go ahead. Ritesh, I have one question for you.
Yeah, sure.
It is very interesting to see that you have reduced so many allergens and then also rebalance the protein. But my question is, have you checked the post-harvest storage efficiency, or maybe the you know because so many things have been changed, and soybean has very less shelf life. So whether the shelf life has been increased, reduced, or it is same.
Yeah, so we did initially the germination test after storing it for like one season, and that didn't affect it a lot. And now with these multiple locations, that's another aspect we be going to test, like how this protein levels will affect like seed germination and other traits. Yeah.
Okay.
Yeah, that is an important fact. I agree, because if we are knocking out some useful storage proteins,
Yeah.
and it may affect the germination or even the growth in the future. But
Yeah.
so far, they grow very normal as compared to the wild type, because
Yes.
we keep these seeds for one generation and then take them to the next generation. So they are healthy and good enough to grow in the field.
So the germination efficiency was same.
Yeah.
Okay. So that's very interesting. Uh, okay, so if there is no question from audience side, I would like to profusely thank Dr. Ritesh for sparing his time in the morning itself. I think it must be around 7:30 for you in the
Yeah, now
morning.
it's 7:45. We
Yeah.
started 7.
Yeah, yeah. Okay. So thank you so much. Whenever you visit next time to Delhi, please do visit us. We'll be happy to
Yes.
host you.
Thank you.
Yeah. Thank you, Ritesh. Thank you.
Yeah, bye.
Okay, for online participants, tomorrow we will revert back at around 10 a.m.